Good morning, everyone. Thank you for joining our final bite-sized science seminar of 2020. For those of you who don't know me, my name is Mara Jean Tilley, and I have the great privilege of heading up the Garvan Research Foundation, which is the marketing and fundraising arm of the Garvan Institute of Medical Research, and I'm delighted to be your MC today. We'll be joined by Garvan's Executive Director, Professor Chris Goodnow, who will take us through what has been both a very challenging but highly innovative year for Garvan and indeed for medical research right across the globe. Before we get started, I'd like to pay my respects to the Gadigal people of the Eora Nation, the traditional owners and custodians of the land on which the Garvan Institute and St Vincent's Hospital are located. We recognise their continuing connection to land, waters and culture, and pay our respects to the elders past, present and emerging. We also warmly acknowledge the Sisters of Charity and our precinct partners here in Darlinghurst, particularly our frontline healthcare workers who've been at the coalface of this most unprecedented year. Thank you to you all. In terms of today's session, you're going to hear from Chris on our 2020 scientific discoveries, and then we will move to a live Q&A. If you have any questions that arise in your mind throughout the session during Chris's talk, please pop them into the Q&A function at the bottom of your screen. Alternatively, you can wait until we are open for the Q&A. If you would like to ask questions anonymously, please feel free to do so. If you'd like me to acknowledge your name, please do enter your question with those details. Garvan's success is dependent on two key aspects: our incredible staff, our scientists, our clinician scientists, and our professional services teams, and our Garvan family. You, our donors, partners for the future, corporate contributors, volunteers, advocates, ambassadors. We're extremely privileged to have so many of you here with us today, and we hope that next year we'll be doing this event in person in the Garvan Auditorium, as well as continuing an online seminar series for those individuals who live interstate or overseas. In these testing times, it's your support, encouragement and engagement that has allowed Garvan scientists to continue their critical work to investigate the underlying causes of many of the major diseases impacting our community, including COVID. I'd like to encourage you to consider making a donation today if you feel thus inspired by this session. I appreciate many of you have already given so generously this year, but if there is anything else you can do, we would be forever grateful. Or if you'd like to make a donation in celebration or memory of a loved one, you can visit the Garvan website and do so via our range of holiday gift cards. Now to the star of this morning's seminar, Professor Chris Goodnow. Chris is a world-renowned immunologist who joined Garvan in 2015 as Deputy Director and took over the role of Executive Director in 2018. His research is focused on understanding the cause of immune disorders to develop more effective personalised treatments. Scientifically, he's best known for integrating molecular genetics and genomics with immunology and autoimmunity. In 2020, Professor Goodnow has led Garvan's coordinated response to the COVID-19 pandemic. And under his leadership, our researchers have joined forces across disciplines and different fields of expertise to tackle COVID-19 from every angle we can and contribute to this global effort. Meanwhile, we've never lost sight of our pioneering research in cancer, in immunity, healthy aging, genomics, and epigenetics. Please join me in welcoming Chris. Good morning, Chris.
Morning, Mara-Jean. Morning, everybody. Wish we were all together in the auditorium. It is what it is, but I guess one of the upsides with being on Zoom is that many people are able to join us that might otherwise not. So swings and roundabouts.
Thanks, Chris. And look, it's been an epic year. How has 2020 impacted Garvan's research compared to other years?
It's been a tough year for everyone, both throughout Australia and around the world. At Garvan, all of our researchers have really risen to the challenge. On the one hand, we've got all of our research, and I'll walk through shortly some of the amazing achievements that our people have done. But on top of that, all of our researchers and our professional staff, as COVID broke at the start of the year, said, look, we think we have extra to offer here. And so on top of all of their regular research, so many of our researchers jumped in remarkable ways to help with the COVID-19 effort, locally and globally. And I think that, to me, has been really one of the spectacular things to see and to be a part of. Now, I might actually go to the slides if I could, and we've got our very expert professional, Joseph, who will be driving the slides for me. And because I really wanted to share with everybody just a few of the amazing achievements that we've been able to have, give you a bit of a sense of what's been possible despite all the challenges of COVID. Now, obviously, during COVID, it has been harder for people to do their research, but it is amazing, actually, the extent to which people have been able to balance working from home. Our IT department is absolutely brilliant, and so they got us all working as capably or more capably wherever we were online, analysing the huge amounts of data that we all generate in the lab. And at the same time, we had very well orchestrated teams that were able to function as A and B teams in case that there was an outbreak, which thankfully in New South Wales we've been able to do incredibly well. So let me tell you first about a little bit about our two examples of how the team has, excuse me for the hospital next door. One of the wonderful things about being here on the St. Vincent's campus. Constant reminder of why we're here. So let me tell you two things about how our researchers have contributed to the whole COVID effort and basically pivoted. So one of the first examples is, as you know, we've got really some of the most advanced genomic sequencing technologies in the country and in the world here at the Garvan in the Kinghorn Centre for Clinical Genomics. And one of the people that leads a new kind of sequencing technology called nanopore was Ira Devison. And so when COVID started, Iris and his team said, well, what's going to be crucial for contact tracing is to be able to sequence the whole genome of the coronavirus as quickly as possible and be able to provide that back to the contact tracers so they know who got the virus from whom, because that reveals a fingerprint. Well, last week you may have seen in the news, but the published work from Ira and his team in collaboration with the Kirby Institute at University of New South Wales and New South Wales Health Pathology published a really landmark paper showing that this nanopore sequencing, which is a kind of new kid on the block, could turn around in 4 hours a complete sequence analysis of someone's COVID infection and feed that back into New South Wales Health Pathology so that contact tracers could say, for example, as we saw a couple of weeks ago in Sydney when we'd had no cases at all, and then suddenly we had a person who was working at 2 of the quarantine hotels came up positive, and the question was, where had that person got the virus from? And what sequencing of the coronavirus as quickly as possible could do was to quickly tell the contact tracers that this was not community transmission. This was had come from aircrew on United Airlines who were staying in one of the quarantine hotels. So a fantastic achievement. Joseph might want to go to the next slide if we could. Thanks. And we were just thrilled to see that this was picked up by all of the different news media. I think it is one of the great success stories of the whole COVID has been New South Wales Health's contact tracing, and we were just thrilled that Garvan could be pitching in even just a small way to make that contact tracing faster and more cutting edge. And so a great example of new technology, but also a great example of how we don't live in an ivory tower here. We partnered with Kirby Institute, with New South Wales Health Pathology. We just got the job done, and I think the team collectively got the job done magnificently, and we're all the beneficiary for it. I might go, well, while we're on that slide, actually, if we could go back one, Joseph, sorry. The other story which I wanted to update you on, we have a terrific immunology here at the Garvan, and Daniel Christ's team here applied all of his know-how about antibodies and how you could produce antibodies in the lab to bypass the vagaries of the natural human immune response and engineer antibodies as a way of protecting people potentially from infection as an alternative or a complement to a vaccine. And Daniel's team has done a terrific job over the course of this year. He's got some extremely promising antibodies that we hope will go into the clinic next year. And in fact, this week he's submitted the first paper describing the antibodies, both their potency against the virus and the structure of those antibodies. So really, again, remarkable achievement in an incredibly short amount of time under enormously difficult working conditions. So two great examples out of more than 14 that have happened across the Institute where our researchers have been pitching in on the COVID effort. Now, if we could go to the next slide. And, you know, the key point which I mentioned before was that all this COVID effort is layered on top of all of our teams working as hard as ever and achieving more than ever in all of the areas that we care so deeply about here at Garvan. And one of those, of course, is cancer. And as one beautiful example, we were all thrilled a month or so ago to see that our very own Dr. Angela Chow was named the 2020 Outstanding Cancer Research Fellow by the Cancer Institute in New South Wales across the whole of the state. It's an annual award recognising an early career researcher who's demonstrated exceptional progress. And Angela is, in many ways, for me, an embodiment of exactly the vision of the Kinghorn Cancer Centre and Garvan and the St. Vincent's campus, and also the interaction more broadly, because Angela is a clinician trained in oncology. Her particular focus is pancreatic cancer, but she's trained in the genomics of pancreatic cancer as part of her PhD and postdoctoral work here at the Garvan with the Australian Pancreatic Cancer Genome Initiative. And again, we're not in an ivory tower. Angela also practices at Royal North Shore Hospital. So it's a beautiful example of how we're able to upskill the whole health system, but it all hinges around our ability to support brilliant young people like Angela to do it all, to be at the very cutting edge of genomics, to work on one of the toughest nuts to crack, pancreatic cancer, and to be a practicing clinician all at the same time. That's no mean feat, and it takes a community to pull it off and do it well. And Angela's a good example of doing that. Now, if we go to the next slide, one of the other great achievements in cancer this year was a paper that was 7 years in the making, a huge study on the part of Akadi Cipioni here at Garvan, a senior research fellow working with David Thomas for the last 7 years, trying to understand one of the biggest problems in cancer across all different cancer types they've been working. And that was when someone goes on to chemotherapy, why is it that the cancers are so good at evading the chemotherapy and becoming resistant to it? As you know, that is one of the big problems that we have, and it's becoming a bigger problem as we get better and better as a medical research community at coming up with targeted treatments for cancer, we are discovering over and over again that the cancers get pretty clever at coming up with workarounds for those targeted treatments. And so what Akani and David and a brilliant team of collaborators did was to use all the tools of genomics and cancer profiling across a range of different kinds of human cancers, both with primary tumor samples in people on chemo and in animal models, to discover that what the cancers have is a circuit that, when they're under stress from going on a particular chemotherapy, they turn on a genetic circuit that allows them to create mutations across their whole genome, and that increases the likelihood that one of the daughter cells in the cancer has come up with the workaround to evade whatever the chemotherapy is. So this is a whole new circuit. It's actually a well-defined circuit for cancer growth and survival, but the real breakthrough was connecting that circuit to a way for the cancer cells to come up with an evasion strategy, much in the same way that bacteria have an evasion strategy when we go on antibiotics to come up with antibiotic resistance. And knowing what that evasion strategy is the crucial next step towards coming up with a drug or other therapy that we can actually close off the workarounds for all sorts of cancers. So a beautiful example, I think, of groundbreaking work that has immediate clinical relevance and draws on all of the expertise that we have in cancer, in genomics, and in large data science. Now, if I could go to the next slide, please, Joseph. Now, breast cancer has long been one of the major concerns for all our researchers here at Garvan. Going right back to Rod Sutherland and his groundbreaking work with his team connecting the estrogen receptor with the machinery for cell division. But this year, our team, and particularly a team led by Alex Swarbrick, but working with our Garvan Weizmann Center for Cellular Genomics and others, applied cellular genomics, a whole new technology that's revolutionized the way we can look at cells, to one of the poorly studied but absolutely crucial parts of breast cancer. You know, for the most part, we focus on the cancer cells themselves, but they live in a milieu surrounded by all sorts of supporting cells that make a fundamental difference to how well the cancer cells can move and escape and spread around the body, and how well they respond to chemotherapy or immune therapy. And what Alex and a big team were able to do was to discover 4 new cell types and the whole gene expression signatures within those cell types that are stromal cells, as we call them, that support and control and create the tumor ecosystem around breast cancer cells. So a huge step forward from looking at what the cancer looks like down the microscope to actually seeing what the cells are doing and how they're using their genomic instructions to support the cancer, and how we can then use it to counter the cancer. Now, if I could go to the next slide, one of the other great achievements was by Joanna. Now I'm going to mispronounce Joanna's name. Joanna Achia-Kaweska. And Joanna is a senior fellow working with Sue Clark's laboratory here at the Garvan. Again, working on breast cancer, but coming at it from one of the other key problems within the cell. And that is that every cell in our normal cell in our body has a full copy of all of our DNA instructions to make any cell in our body and to make our whole body. And the crucial thing is that each given cell needs to shut down and most of those instructions and just go to the parts of the genetic encyclopedia, if you like, or the genetic cookbook needs to go and just get the right bits of instructions and lock away all the others in compressed, filed away folders, if you like. And that involves a lot of 3D rewiring of the genetic machinery to open up some books and close down all the others. And what Joanna has been able to do with her team is to figure out how that wiring, that three-dimensional wiring of the DNA genome within a cell is changed in a breast cancer cell as part of how the cancer departs from normal instructions and becomes malignant. To give you a bit of a sense of that, I might go to the next slide and show you a little image of what Joanna what Joanna's discovered is fundamental links that connect different parts of the DNA together so that they are either closed off so that the cell can't read those genetic instructions, or opened up so that the cell has access to a particular part of the genetic library that it should or should not have. Really fundamental work that will tell us an enormous amount about how cancer cells grow and how we can attack them and target them to stop them coming back after chemotherapy. Now, if we go to the next slide, you know, the overarching goal of medical research, or one of the goals of medical research, and one of the great achievements in medical research, has been that we all live so much longer now than we ever did before. My kids, who are in their 20s now, can expect to live over 100. But there's no point in living to over 100 if you're not well and healthy for all of those innings. And that means we've really got to get on top of some of the chronic diseases that become more frequent as we get older. And there's none more sinister in many ways than dementia. And so one of the great achievements that happened this year was led by Dr. Catherine Samaras, who heads our healthy aging theme at Garvan. And what Kathy illustrates is, again, how you can break down the barriers between the clinic and the laboratory, but also between different medical disciplines, in this case between neuroscience of dementia and the endocrinology of diabetes. And what Kathy and her team did was to publish a very important paper, again requiring years and years of painstaking work and crucial support from the community as well as from the government, to show that in people with type 2 diabetes, a drug that's very safe to use and widely used and inexpensive does indeed slow the cognitive decline and slow dementia. And so on the strength of that, Kathy has now, with her collaborators, won a very large grant from the National Health and Medical Research Council to run a full randomized controlled trial of this drug, metformin, in people with type 2 diabetes to provide the full evidence base that could ultimately lead to many, many more people who are at risk of dementia, with or without diabetes, potentially benefiting from metformin. So really a great example of being able to take the long view, 6 years in this case, this study was in the making, but obviously with immense benefit and you know, tackling one of the big problems. Now, while we're on the topic of diseases of aging, if we could go to the next slide, my own team, and again, it's a huge team effort involving, again, collaborators at many hospitals across Sydney, in Adelaide, and the Garvan Weizmann Centre for Cellular Genomics, the experts in genomics and cellular genomics at UNSW and many other places. What we were interested in is autoimmune diseases and what causes them and why do they become also so much more frequent as we get older. For example, rheumatoid arthritis, which affects over 1% of particularly women over the age of 70. And to cut a long story short, in a paper that was published in Cell early this year, the team was able to show that these cells that cause people to become very sick with an autoimmune disease that affects the blood vessels very seriously as we get older, that the rogue cells had acquired mutations, the same mutations that you find driving cancers, lymphomas and leukemias in particular. But in this case, the mutations were not creating a cancer, they were creating a rogue cell that was making damaging antibodies that then caused a chronic disease of aging, autoimmune disease. And we've got, again, another fantastic little video, again, from our wonderful medical science illustrator, Kate Patterson, which I might show you now. And so here we are on the surface of one of these rogue cells, and we're looking at the antibodies that these rogue cells are making, immunoglobulin G. And what we're seeing is that the antibodies on the surface of the cell are acting as a receptor that are sensing a normal part of our blood plasma, immunoglobulin, and normally this would stop these cells from making that antibody, because if they do secrete these green antibodies, they will damage the blood vessels and the kidneys and the nerves. But in these rogue cells, they're actually being stimulated by the self components because of the mutations that have arisen in their genetic wiring. So again, we're really being able to see for the first time the cause of a chronic disease. And of course, that sets up a pathway towards a targeted prevention or cure. Now, I might go to the next slide. And again, this is to me one of the most satisfying examples of the light on the hill of how all of this basic science across many disciplines, cancer, diseases of aging, young diseases, diseases of bone, diabetes, neuroscience, etc. What's the advantage of having all of this under one roof, and how does this genomics actually really come home to make a difference for people? The light on the hill, if you like, is this little girl that you can see in the image here, Carla. Carla first came to Westmead Children's Hospital when she was only 1.5 years of age. She had a very early onset of rheumatoid arthritis, as I mentioned, a disease that normally becomes quite frequent in women over 70, but Carla was just a baby. She also had another autoimmune disease that was life-threatening, that was destroying her liver. And her brilliant pediatric gastroenterologists and rheumatologists were able to control the liver rejection, but they could not stop the joint damage, her arthritis, to the point that she was no longer walking and was in immense pain, and was going on the journey of trial and error of different drugs, when a team, who you can see some of the individuals here at Garvan, sequenced her genome and found the cause of why she had such early onset autoimmune disease. And it was a single gene defect. One of the important breaks on the immune system, one checkpoint, as we call it, was broken. But the really exciting thing was that broken checkpoint could be replaced by an approved drug that's approved for kids and adults with rheumatoid arthritis, but actually only works in 5 or 10% of people with rheumatoid arthritis. But the science told us that it was very likely that Carla would be an exceptional responder. And so her team at Westmead Children's was able to use that information to switch her on to the drug, abatacept. She's now 2.5 years out, and you can see her here with her mum visiting Garvan, she is now a energetic, bright as a button young 7-year-old, and just having a ball with no problems with rheumatoid arthritis as a result of putting the science and the clinic all together. I wish we could do that for every disease of the young and the old, and that is really why Garvan's here, and that's why your support is so crucial. So really I might wind up there with just my last slide, which is just again to say thank you. Garvan's team, does a brilliant job putting the science and the clinic and the big data and everything else together. They do a great job of winning grants from national, local and international funding bodies. But as you probably know, the grant-giving bodies usually don't give you the grant until you've already shown that it's highly feasible. So you have to have the work almost half done before you get the grant. And that's where philanthropy really comes in. Your gifts allow us to allow these projects, some of the ones that I've described to you, that often are 6, 7, 10 years in the making, really groundbreaking things, to get a long way down the runway before they can actually get the government support to take off. And that really is what's special about Garvan, and what's special about being part of the community that you all are. So thank you.
Thanks so much, Chris. Fantastic presentation. Now, just a friendly reminder for anybody with questions, please do put them through the Q&A function at the bottom of your screens. Otherwise, I will hog this time to get all of my questions answered from you, Chris, if I may. Now, the first one is in relation to the COVID-19 vaccine, Chris. I've heard that while the vaccines that are currently being tested might help prevent recipients from getting dramatically unwell with COVID-19, it may not prevent individuals actually getting the virus and therefore shedding it and spreading it to others. So is that true? And if so, what does that mean for the global community as the vaccines are rolled out?
Yeah, it's a great question. You're absolutely right, Mara Jean. There are two things with vaccines. The first one, which is so wonderful, particularly with the Pfizer vaccine and the Moderna vaccine, no one expected 94% protection against getting ill if you're infected. And that is a huge step forward. And when you step back, when I saw that news back a month ago, I got up early in the morning and checked my phone, and I just thought, wow, they designed that vaccine, that new kind of vaccine RNA, in literally the design was locked down and went into production for clinical trials in March, when the sequence of a new virus only became available at the start of January. And the only reason that was possible was because of 20, 30 years of research in labs around the world, here at Garvan, many other places, that had created the knowledge base for how you would design a vaccine against a new virus, so that once the virus sequence is available, you could just plug and play. You knew what not to do, you knew what to do. And it is an incredible example of why medical research delivers. Now, the big question is, the great thing is protection from disease is really important, because that means now if we can roll those kinds of vaccines out, and I think then that will decrease the risk that if someone's infected, they end up in ICU, or worse still, that we lose, someone that's very dear to us. And that, obviously is job number one. Now, the second thing we want is to stop the virus spreading in the community, because not everyone will be able to get the vaccine for one reason or another. And so the people who can't be given the vaccine, or the 6% who, if they're given the vaccine, aren't protected, we can't have the virus running riot through the community where 94% of people are protected, but the other 6% are now getting hammered. That's not going to be acceptable to anyone. So what we really need is to know is which of these vaccines are not only going to stop people from being protected, but will actually stop people from getting the virus and passing it on asymptomatically. And we've certainly seen that's been the big problem with this virus compared to SARS back in 2003. With SARS, there were no asymptomatic spreaders. So it was actually easy to shut down the outbreak and limit it to 8,000 people around the world. But the reason why this cousin of SARS, SARS-CoV-2, has been so hard is because the majority of people are either mild or asymptomatic spreaders of the disease. And so we don't want the vaccines to make that even worse. So there will be many studies that will roll out over the course of the next 6 to 12 months to give us a sense of how much the vaccines are stopping the spread. But at this stage, it's only guesswork, unfortunately. And so that will mean things like international borders. The vaccines won't really solve that problem for us in the short term.
Thanks, Chris. It really emphasises how fortunate we are in Australia that we've managed to handle it so well. So it sounds like there's still a long road ahead. We've got a question from George. Aged care has received a lot of attention this year. Professor Samaris's work is exciting. Is there a program of work aimed at helping aged care providers improve their systems of care by linking it to the various research streams at Garvan?
Oh, wow, that's a very interesting idea. We don't have an across-the-board strategy along those lines, and that's an really interesting suggestion. We have, for example, the Garvan fracture risk calculator, which is focused particularly in osteoporosis, which is such an underdiagnosed condition. Often people don't know they have it until they break a bone. And so the team here at Garvan has worked for many years and is continuing to add genomics now to this, to essentially have that as something that's certainly on every GP's computer and desk as a way of identifying which of the people they care for might be at risk of a fracture and, stitch in time. But the more general thing with aged care is an interesting idea and something that we're in the midst of doing our strategic plan for the next 10 years, and that actually is a great suggestion.
Fantastic. Thanks, Chris, and thanks very much, George. Chris, I caught up with one of our supporters yesterday and they put me on the spot with a very difficult question, and I'm now going to throw it to you because I think I only answered it partially. So, through the pandemic, we've seen an absolute global revolution and activation of medical research. And I think that it's fair to say that the vaccines that are currently being rolled out have been created far quicker than anybody could have expected, albeit, building on the work of many, many scientists over many, many decades. So the question I got asked yesterday is, how is it that we've produced multiple new potential vaccines in a matter of months, but we haven't yet solved cancer, autoimmune diseases, what else did they say? Or the diseases of aging?
Yeah, that is a great question. And it is the core question for every researcher such as myself. And that the short answer is, well, in the case of the, as I mentioned before, in the case of the vaccine, the problem had been reduced down because of all the research that's been done. The problem had been reduced down to the point that we knew exactly which part of the virus we needed to make an antibody to. We knew exactly, had a pretty good idea of how the different approaches to elicit antibodies safely in a vaccine. And so it was just a matter of engineering to implement that. There were no, the biggest known unknowns, and there were some substantial ones, the biggest known unknown for this class of viruses was, would antibodies actually do the job of protecting people from getting sick? There was even a substantial probability that antibodies might make you get sicker. So that was the biggest risk, and we didn't know the answer to that. We do know the answer to that now, and that's actually one of the most important things of these first couple of clinical trials, is it tells us antibodies do the job and they don't make you sick. So that's narrowed the degrees of freedom even further. It's just a matter of how much antibody can you elicit and for how long. When we get to cancer, at the other extreme. So if I back up, back at the beginning of the 1970s, Richard Nixon, as President of the United States, decided to declare a war on cancer. And he ramped up, they created the National Cancer Institute as part of the NIH, and ramped up a huge amount of funding against cancer, with the view being that if you threw enough resources at it, cancer would cease to be a problem in 5 years. This is in 1970. It's still with us. But having said that, we are now the beneficiaries of everything that was set in motion from that war on cancer. 80% of people with breast cancer, for example, will be cured in their first cycle of surgery and chemotherapy. 95% of kids with leukemia will be cured. There have been great strides, and there continue to be great strides. The checkpoint inhibitors for melanoma have been a complete game changer in terms of immune therapy. But why did it take so long? And it's very interesting to go back to 1970. In 1970, we did not know what caused cancer. We did not know that it was mutations in the DNA. In fact, to sequence 6 letters of DNA, John Shine was doing his PhD at that time before he became director of the Garvan Institute. It took him 3 years to sequence 6 or 8 letters of DNA at that time. And so everyone was convinced in the research community, well, not everyone, but a substantial number of people, that cancer was caused by viruses. Now, some cancers are, cervical cancer being, that with papillomavirus being a very good example, but most cancers are not. Melanoma and lung cancer, it's just this huge burden of DNA mutations that are created by exposure to ultraviolet light over many years. Me out there on a surfboard through teenage years with no sun protection. Lung cancer, like my father, smoking away like a chimney all of his life. But in 1970, we did not know that. And we didn't know, it was only in the mid-1970s, as a result of the war on cancer and the direction of things, that scientists identified the first mutated cellular oncogenes, as we call it, normal genes that have been corrupted by a mutation to drive cells to grow out of control. So the bottom line, it's a long answer, but to a very, very central question, which is that resources are important, and they're one of the limiting factors. But the other 2 limiting factors are conceptual. What do we not know? What are the unknown unknowns? And in diseases like cancer or autoimmune disease, there are so many of them. And we're progressively shifting more of them into known unknowns or even known knowns. And then the third crucial limiting factor is technology. And it gets, again, that example with the war on cancer is relevant. It took so long to just sequence a very small segment of DNA for the first oncogene to be described, the cloning of the first chromosome translocation activated oncogene, which was c-Myc, depended on technology that John Shine invented after his PhD to clone genes in bacterial plasmids in the mid-70s and late 70s, and to do what our team, what Angela Chow has done as part of her PhD, to look at the complete genetic aberrations of someone's pancreatic cancer genome. That only became possible in the last 10 years with massively parallel whole genome sequencing, developed, for example, by a company that I had the privilege of being the founding scientific advisor for, Illumina. But we still have an enormous amount still to go. The examples that I gave of Alex Swarbrick with single-cell genomics, we can do things this year in terms of genetic profiling of single cells that are leaving behind what we used to do in terms of looking down the microscope at cancer cells. But in 5 years or 10 years' time, we will look at what we can do today and go, well, that wasn't even technology. That was a bicycle. So funding, very crucial. People and concepts, absolutely the crucial thing, and technology. And so those are the three pillars of Garvan.
Thanks, Chris. I wish you were with me yesterday when I tried to tackle that. That was a fantastic answer. We've got another question coming through from Sandra. Hello, Sandra. Is the link between metformin and dementia due to its chemical structure and an effect of it in the brain, or due to a reaction of metformin in some other way? Great question.
That is a great question. This is really a Kathy Samaras question. She knows more about metformin than pretty much anyone on the planet. The answer that she gives when we and others have asked that question is multifold. That metformin does seem to have a number of mechanisms of action. One that's particularly well accepted, and that may explain the connection to dementia, but that's within a cell. Another is by modifying how much glucose is in the bloodstream, and therefore how much of the glucose gets oxidized into what we call advanced glycation end products, which potentially damage the blood vessels or even cross the blood-brain barrier. There's that. So I guess the answer is, the short answer is we really don't know. The first step is just showing that it does work, and her data so far shows that's the case, but now needs to be confirmed in this big randomized trial that she's just starting. And yes, the parallel question is, how do we learn from that, the mechanism, so that we can extend it more generally to dementia? Because as you probably know, for both dementia and Parkinson's disease, those come back again to this question of enormous numbers of unknown unknowns. The conceptual landscape of exactly what drives the development of tangled proteins in our brain, which is cause, which is effect, which is feeding on each other. Again, this is where we come back to genomics as being our secret weapon, not so secret weapon, because it helps to disentangle which is cause and which is effect in a way that almost nothing else can in the laboratory or the clinic.
Fantastic, Chris. Thank you. So, Sandra, stay tuned. We'll keep reporting on the outcomes of that clinical trial as it gets started. Great question. Thank you. Now, we are running out of time, but Chris, I guess as we go into summer and our Garvan family are finally getting to catch up with loved ones in other states and leave their home location, etc., what would your advice as an immunologist be for everyone to stay safe and ensure that we don't follow other countries around the world, such as the UK, which I believe we've got London in stage 3 lockdowns at the moment?
Yeah, well, I'm torn because as a member of the community and as a father, um, uh, and having my daughter just arrived from Melbourne, we haven't seen her all year, and so I think as a person, my answer is make hay while the sun shines. Um, at the moment, thanks to our governments, state and federal, and thanks to the whole community, we have got zero community transmission. Huge achievement, and a huge collective achievement on the parts of all of us to give up a little bit of our individual rights to the community as a whole for the greater good. And so I think we do need to be able to reap the rewards of that by being able to get together with people we haven't seen all year and not by Zoom.
Absolutely.
So that's what I'm going to be doing. But I won't be shaking hands. I'll still do my elbow bumps. So that the reality is that 3,000 people a week come in from overseas who are desperately trying to come back home either for Christmas or to get back to Australia or have crucial new jobs to take up here. A certain fraction of those are going to be bringing the virus, and quarantine's never 100%. 99 point whatever percent, but not 100. And so we will have situations like the one I mentioned a couple of weeks ago where it leaks into the community. And obviously the rapid coronavirus sequencing technology is one extra piece to the New South Wales Health team to speed up the contact tracing. We need to get right behind our New South Wales Health team. If they ask us to do things, if they ask us to get tested, we need to get tested. That's the single most important thing. So if you have a sore throat, you wake up, go get tested and be proud of the fact that when the test comes back negative, you've done the right thing. And don't feel like you wasted people's time or resources. No, absolutely. You should feel very proud to go off and get that text in the middle of the night saying you are negative. And thank you, because you have now done your bit to keep us all safe.
Fantastic advice. Thank you, Chris. And Chris, one last question, just very quickly. Where will you be surfing this summer?
Well, it just depends on the angle of the swell. It looks like we'll have a cyclone swell for Christmas. Unfortunately, that's going to be at the expense of the Fiji Islands, but with a Category 5 cyclone, but probably at Manly.
All right. Well, everyone, keep your eye out for Chris. If you spot him, please do say hello. Thank you, everyone, for joining us. As Chris has said, Zoom is very useful. We do miss seeing you all in person though, and we're really grateful that you've taken time to join us this year through our virtual seminar series. It would be remiss of me on this occasion not to acknowledge and thank the team behind the scenes, Priyanka and Joseph and many, many others who do incredible work all year long to help us be able to bring events such as this to you so that we can share the research that your support is directly enabling. We're really grateful for everything you've done for Garvan in 2020. If you do feel inspired by today's session, please do consider making a donation. Please also share the recording of this seminar with anyone in your family or friends group who you think might find it interesting. That link will go out in the next couple of days, all going well. And let's all stay safe and sane and well over the summer period, and hopefully we'll be catching up in person, albeit still bumping elbows, no shaking hands in the new year. And we really look forward to welcoming you back to Garvan in person and continuing our sessions online as well. Thank you, everyone. Please take care. See you soon.